Okay, well, good morning, everyone. I'd like to begin by thanking the program committee for giving me this opportunity. It really is a privilege to get to talk to you today about imaging in an era of multi-scale neuroscience. And as Angie said, I'm going to be talking about this from the perspective of somebody who develops tools. So I work on techniques for acquiring, reconstructing, and analyzing MRI data. And it's almost a bit of a cliche to start by showing you this slide. But it really is the case that one of the key challenges we face in neuroscience are a series of challenges of scale. In the spatial domain, structure and function relate to each other over something like 9 orders of magnitude. I'm showing you here 6, from chemical signaling that happens at synapses all the way up to macroscopic structure. And of course, in some senses, it's very tempting to think that the way that we should tackle this problem is to start on the left and slowly move toward the right until we build up an understanding of the brain as a whole. But the sort of fallacy in that thinking is really nicely described by this quote from a paper by Philip Anderson, where he said, "At each level of complexity, entirely new properties appear, and the understanding of these new behaviors requires research which is as fundamental in its nature as any other." Now, he was a physicist talking about biology, but the same kind of concept really holds within neuroscience as a microcosm. Although we certainly need to understand what's happening at the microscopic scale, there are certain aspects of brain function and health that we're only going to understand if we look at the brain as a whole. And at the end of the day, one of the key challenges that we face is actually to link across these scales. Now, of course, there are also challenges of scale and time, where we have neuronal firing that's happening on the order of milliseconds, learning processes that happen over hours or days, and developmental processes that can take years. And so we're faced with this enormous domain that we need to span in neuroscience. And over the last few decades, neuroscientists have been really adept at creating a sort of dizzying array of tools that allow us to span this domain. But I think increasingly people are beginning to understand that maybe the major leaps forward are no longer going to be made by filling in the remaining gaps in this space, but instead by trying to link across different techniques, different species, and different scales. Now, there are other challenges of scale that we maybe don't describe as such all the time. One is the scales of people. So we tend to be doing imaging experiments and cohorts that are very homogeneous and relatively small. But if we're going to have the biggest impact in neuroscience that we could with neuroimaging, we need to be able to work in populations, as well as be able to characterize individuals in great detail. And so today I'm going to be talking to you about how I think neuroimaging can address these issues. And I think one thing that's important is, although we sometimes think of ourselves as using human imaging techniques, particularly MRI, as sort of sitting off at one end of these scales, there is something really powerful about the techniques that we have that enables us to delve into some of these different aspects of scale, which is that neuroimaging is very, very flexible. We can use it in a really broad range of different neuroscience areas, including characterization of small tissue samples or causal experiments in rodents. We can look at patients, we can look at populations, and there aren't actually many tools in neuroscience that have that flexibility. So one of the things that I've become very interested in then is how we can take advantage of that flexibility to link across scales, to relate what we're doing to the scales that some of these other techniques in neuroscience can give us. So I'd like to begin by talking about temporal scales, and in particular how we can leverage the temporal domain to improve fMRI data. As you all know, in fMRI, we identify activity based on temporal covariation, whether it is with a voxel in comparison to a task, or if it's voxels relative to each other. And this means that the fMRI data is fundamentally a sort of a spatiotemporal beast. It has spatiotemporal structure. Voxels carry temporal information about each other. And so although we may often think about this as a spatial mapping technique, the temporal domain is actually absolutely critical to what we're doing. And from that perspective, you can see that the dimensionality of fMRI data is actually quite problematic. We might have, if we were to unwrap an image into one long vector, we might have 200,000 voxels in an imaging experiment, but only 200 time points, because each of those voxels carries a burden of temporal acquisition time. And so from this perspective, you can see that the fact that it's the temporal domain that is encoding brain activity means that we're, in a sense, quite information poor as long as we stay in this kind of a domain. And so there's been a lot of interest in the last 5 to 10 years in developing fast fMRI techniques, where we're trying to increase the density of temporal sampling. One way of looking at the benefits of this is that it increases the degrees of freedom in your experiment, and that brings improved statistical power. So for example, here in this resting state scan, you can see that a threefold improvement in the temporal resolution enables us to pull out brain networks with much higher statistical significance. Now, the main technique that people have, the main technique that seems to have kind of gained prominence in our field over the last 5 to 10 years, largely in large part due to the efforts of the Human Connectome project, has been simultaneous multi-slice imaging. So the idea here is that we can speed up acquisition if we can acquire slices simultaneously. We can move through the brain more quickly by doing this. So if we were to naively reconstruct an image with multiple slices simultaneously, they would be overlapped. So what we need to do is to be able to separate them out. And the way that we do this is we take advantage of a bit of hardware, which are the sensors that we use in fMRI, or the coils, which are distributed around the head. And because they're distributed around the head, they confer some amount of spatial information. And so with some clever maths, you can separate these slices out and get faster fMRI. One example where this can help, that's not simply down to statistical significance, is that it can enable us to do new types of analyses and identify new information that we can't easily do using current data where we have hundreds of time points. So for example, if we can expand to now thousands of time points, we can do things like temporal ICA, which look for temporal independence rather than spatial independence in terms of characterizing networks. And what this enables you to do is to identify networks that are not overlapping in space and which are non-stationary in time. And this is a really nice example from Steve Smith, where he's showing an extended network that you can identify using this approach, which actually corresponds very nicely to a working memory task. Now, of course, these techniques have their limits. If you accelerate to a very large degree, there are predominantly two types of artifacts that start to crop up that prevent you from kind of gaining further benefit. On the right, I'm showing you noise amplification. It may be a little hard to see in the image. Let's see if I can get it over here. In this image here, but there is noise amplification. But if you look at the statistical map on the right, you can see very clearly that the statistical significance at this acceleration factor of 12 is reduced compared to an acceleration factor of 8, and that's down to noise amplification. Similarly, on the left, you can see another example of an artifact, which is where slices haven't been properly separated. And so you have artifactual activity that actually belongs in the occipital lobe, but which has been incorrectly placed into other slices in the white matter. So in my group, we're interested in thinking about, are there ways that we can then get further accelerations, which don't rely on coils the way that the simultaneous multi-slice techniques do? And this is work from Mark Chu in my group, where we're interested in sort of exploring what we can do if we move away from one of the classic tenets of image reconstruction, which is that every time point is reconstructed independently. So in other words, each EPI volume is reconstructed without knowledge of the other volumes in the time series. And yet we know that voxels carry information about each other in time. So can we leverage the temporal domain somehow in the reconstruction in order to gain further benefit? And what this really builds on is something that we take inspiration from in data analysis, which is that fMRI data dimensionality is lower than the number of time points. And that has important implications about the way that we can represent the data. Essentially, we know from analysis that we can represent data using a series of spatial maps and associated time courses. This is essentially exactly what the GLM does, and it's what methods like ICA do. So for example, in ICA, if we have our data matrix of space by time points, what ICA does is to represent that as a number of components, each which has a spatial map and an associated time course. And crucially, it represents a number of components that is relatively small compared to the data itself. So even up to 100 components still has fewer total points in this representation of ICA compared to the original data set. So because there's fewer points that we have to estimate, fewer values we have to estimate in that independent component analysis, that tells us something about the data, which is that we shouldn't have to acquire all of it. We should be able to reduce the total number of measurements that we make when we acquire the data, and still be able to create this reconstruction of ICA on the right. And so this is exactly what we want to do. By not having to acquire as many points in the spatial domain, we can move the burden of that into the temporal domain, increasing our temporal resolution. But what we want to do is to do this based only on the observation that we should be able to represent that space by time matrix using a small number of components. So that may sound a bit magical, but it's actually a problem that you're very familiar with, which is the Netflix problem. So what the Netflix problem is when you log on to Netflix, it wants to try and recommend movies to you, what you would like to watch, right? And it has a database of some number of viewers and some number of movies, and it roughly knows how much you like a given movie, right? And so what it's trying to do is to predict from some movies that you haven't seen, so missing data points, is trying to predict the values, how much you would like each of those movies, right? And intuitively, you understand why this works, right? You are not a special snowflake, okay? You are a canonical user of some type, right? You like sci-fi movies, or you like rom-coms, maybe you like both, maybe you hate both, right? The Netflix problem says, all that Netflix has to do to fill in these missing points is to figure out first, what are the canonical types of users, sci-fi, rom-com, et cetera? And second, what type of user are you, right? And so intuitively, that problem is intrinsically solvable because of the fact that there are certain types of canonical users. So this is what's called a low-rank matrix recovery problem in signal processing. And it's the same problem that we have in fMRI, but now replace types of movies with networks. So the way that this reconstruction works is we have two constraints. The first is a data consistency constraint. So we're going to require that where we have actually acquired data points, that our reconstruction is consistent with those. And then second, we're going to include what's called a rank constraint. This is basically a dimensionality constraint. We're going to require that the data can be represented by a number of spatial maps and associated time courses. And this works remarkably well. So if we look at an example where we have a time course at a given k-space location where we have undersampled, so we're only acquiring data at the samples and the circles. Here I'm showing you a retrospective simulation based on real data, so we have truth. And you see that the reconstruction in blue does a remarkably good job of filling in the missing points. Importantly, it's not just interpolating between them, but it's capturing dynamics. So for example, here you can see that it's capturing dynamics in between points that it's actually seen. And the reason why this works is because voxels carry information about each other. That although we haven't looked at these particular k-space points, we have acquired other points at those points in time, and they contain that information. So Mark has demonstrated the ability to accelerate up to quite impressive factors, up to factors as high as 20. You can start to see that it's beginning to potentially degrade a little bit, but it's imparting a clear improvement over what we're able to do from a time-independent reconstruction. Now, those reconstructions were only leveraging the idea that you can describe the data as space by time. What happens if you know something about what these space or time domains should look like? Well, as it turns out, if you, for example, have a task or some other measurements, or you know what a spatial map looks like from a functional localizer, then you can incorporate that into this model very easily. So this is what the reconstruction that I showed you a moment ago, which is essentially building an ICA-like model. And we can simply include another component that is a GLM-like model if we know what a regressor looks like. So the exciting direction that Mark is taking this now is in trying to do integrated EEG fMRI reconstruction. So he's taken simultaneous EEG data where he extracts some time courses from that, and then he builds those into his reconstruction. He's basically telling the reconstruction, okay, you need to describe it with some spatial maps and time courses, but this is a really useful direction to look in, okay? And what this is showing you here is that with the addition of that EEG constraint, he's able to improve the stats. And in this particular example, he's able to push something beyond the P of 0.05 level of significance. And so I encourage you to check out his poster here on that. So one interesting, really intriguing idea that this type of way of thinking about the acquisition and reconstruction raises is the possibility that we could actually really fundamentally change the way that we go about conceiving of our fMRI data. We typically think that we acquire some raw data, we reconstruct some time series, and we analyze it. But if you think about what we're doing, we're actually directly reconstructing the ICA subspace using this technique. So it kind of creates this intriguing possibility that maybe we don't even need to form images. Maybe there might be some good reasons why we would like to actually go from raw data straight to, for example, networks in the brain. Okay, so I'd like to move on now to spatial scales. So this is an area that my group has been working on a lot over the past decade or so, where we're particularly interested in looking at relating diffusion signals to what we can measure with microscopy. Now, many of the concepts that I'm going to be talking about now could equally easily apply to other domains, for example, to relating fMRI to electrophysiology, similar to what I was just describing in Mark's work. And the motivation for this, of course, is that although we're exquisitely sensitive with MRI, we have relatively limited specificity, whereas microscopy techniques are able to glean specificity by virtue of the fact that they are resolving a lot of the structures that we're interested in. Now, of course, that comes along with other issues. So in MRI, we're able to roll it out at very impressive scales. I'll talk about imaging at the level of populations later on, whereas in microscopy techniques, we're working in excise samples. We typically can only work in a relatively small number of samples. So these are really very complementary techniques, and what we want to be able to do is to leverage their relative strengths against each other. So I'd like to begin by talking just very briefly about where are we with respect to spatial scales in vivo. And over the last decade, there have been a number of advances in acquisition techniques like simultaneous multislice, as well as improvements in hardware in terms of coils and high field. And these have enabled us to really push the spatial resolution of diffusion MRI far beyond what we were able to achieve a decade ago. So here, for example, I'm showing you a technique we've been developing, which is a 3D multislab diffusion technique developed by Wenxuan Wu here in my group. And at 7T, he's able to demonstrate 1 millimeter spatial resolution and a real fundamental improvement in what he's able to get from tractography. So using relatively simple seeding techniques, he's able to demonstrate, for example, that in the cingulate bundles you see here, he's suddenly able to depict the fibers as they go into cortex, whereas the exact same protocol of seeding is not able to pull that structure out at lower spatial resolution. Now, of course, as with fMRI data, the current techniques that we have can only be pushed so hard. So we were interested in, once again, going and thinking about how we analyze our data, and can that tell us something about other dimensions that we can use to try and improve the imagery construction. And here, the equivalent of the time domain that I was talking about is Q-space. So Q-space in diffusion it's basically the space of directions that we sample. So we can think of this as points on a sphere. And importantly, each of those points is an imaging volume, and crucially, the signal is smooth in Q-space. So if we look at imaging volumes from points that are near each other in Q-space, they look very, very similar because we only have small deflections in the angle of fibers that we're sensitive to from one point to the next. Whereas if we look at points that are further away in Q-space, we see that the images look quite different. So this has been exploited to great effect by Jesper Andersen at our center, who came up with the idea of Q-space modeling that exploits this local spatial structure. And he's using this for cleanup of artifacts. So he's using this to remove eddy currents from data. And the concept that Jesper developed is building on what's called a Gaussian process, which uses, in a data-driven way, aims to characterize what the local spatial smoothness is, or smoothness in the Q-space domain, rather, and exploit that. What it does specifically is it'll take a number of adjacent neighboring points, these purple points here, and it will use them to form a prediction of a target point, the point in red here. So Jesper is using this for artifact correction. What we're interested in doing is to integrate this into the reconstruction process itself. So for a given target point here in red, we're going to pull data from the adjacent points, and we're going to do this in the context of parallel imaging. So here I'm showing you in-plane parallel imaging. We can do this also with simultaneous multi-slice imaging, where we have, again, missing data points that we need to fill in. And typically, we would fill these in using just the coil information. But just as we use the temporal domain in fMRI to fill in missing data, here we're going to use the Q-space domain to fill in missing data. And this approach is really one of the key things that you need to do in order to make this approach work well is to be sure that the adjacent points in Q-space sample different regions in K-space. So in effect, they are contributing information about those lines that you haven't seen. And again, this seems to work remarkably well. So Wenxuan, there's a poster that I'll be presenting this afternoon, actually, where we're demonstrating how well this works. So this is a simulation that we did in order to really demonstrate the sort of extreme example of this, where we've purposely set up a simulation that aims to do something that parallel imaging cannot solve, which is the situation where you're accelerating more than the number of coils that you have, right? And so we know that sense, which is a parallel imaging reconstruction, should not be able to deal with this problem. It's completely ill-determined. But if you look at the Gaussian process sense on the right, you see that we're able to reconstruct actually a very nice image. And when you look at the error on the bottom, it's purely noise. So if we look at a more realistic setting, so this is an actual experiment in a real subject, using simultaneous multi-slice data, here we're accelerating to a very high degree, a total factor of 12. So we're simultaneously acquiring 4 slices and accelerating in plane by a factor of 3. And for those of you who are familiar with this kind of a problem, it's not surprising, again, that the sense reconstruction fails. We've just pushed it too far, whereas the Gaussian process is able to capture this. So inevitably, we're going to keep pushing forward and forward with the kind of spatial resolution that we can achieve in vivo. But ultimately, we're never going to be able to resolve individual fibers or even the smallest tracklets. So it's really important, actually, in sort of fulfilling the promise of in vivo MR, that we try to relate what we're doing to microscopy. And if we're going to do that effectively, we need an intermediary. And that intermediary is postmortem MRI. It shares common signals with in vivo MRI and common tissue state, and actually oftentimes common tissue to the microscopy that we then go on to section those brains for. So the idea of doing postmortem MRI is by no means new. And in particular, if you have a brain that's small enough, macaque size or smaller, you can fit it into a rodent scanner. And in that situation, relatively minor modifications to the standard techniques that we would use in vivo enable you to get images like this beautiful image from Tim Darby. But human brains don't fit into macaque scanners or rodent scanners. So if you try the same trick and just with minor modifications try to use a spin echo in a human brain on a human scanner, you get absolute garbage out. And the problem is that these scanners don't have the strength of gradients that rodent scanners have. And also the tissue itself has changed due to fixation and death in ways that's very problematic for diffusion imaging. So we need to do something different. And the different thing that we came up with was using a technique called diffusion-weighted SSFP. And specifically, the shortcoming that it aims to deal with is that the time between when we excite signal and when we actually acquire it in a spin echo is very long. And diffusion-weighted SSFP enables us to shorten that time without reducing diffusion contrast. And so after a fair amount of development, we're able to get images that look, for example, like this in a postmortem brain. So this is a 0.5 millimeter scan. It took 5 days, okay, so this is not our standard protocol. But you can get this kind of quality of data in human brains, which was really not achievable before. And those of you who are used to looking at diffusion data, hopefully you're starting to see structures pop out that you don't normally see. And this is really showing the power of what you can start to see when you go to higher spatial resolution. Now, of course, when you start to develop techniques like this, people begin to come out of the woodwork. And actually, the most enthusiastic users we've had of these techniques have been people at other sites, or people who want to scan kind of exotic brains. So we've scanned gorilla brains, whale brains, extinct species like thylacines. This is a really nice collaboration that we had with Greg Burns demonstrating what we can do in terms of just basic neuroanatomical questions. So he's interested in understanding how the dolphin auditory system is modified to enable echolocation. And it's a very interesting story that I don't have time to really tell, but it's basically, it's kind of hijacking a bit of the visual system and wiring it up to the auditory system. And so this is a really nice demonstration of where these techniques might have other uses. One of the key things that we are interested in with these very high-resolution scans is looking at the limits of diffusion tractography. So just to give you one example, there's a well-known effect in diffusion tractography, which is called the gyral bias, where the true connection structure is, as fibers come into a gyrus, is that they have to kind of turn by about 90 degrees to reach the banks. And they turn over a relatively small spatial scale. And so because diffusion MRI is limited in terms of its spatial resolution, right at the boundary between gray and white matter, you aren't able to capture at low resolution those turns. And so you end up with this preference for finding connections at the crown and not along the banks. So this data enables us to start asking questions about that. For example, what spatial resolution would be required in order to capture this? And here in our data, you can see, for example, that at 0.5 millimeter, we capture that structure really nicely, whereas at 1 and 2 millimeter, we clearly have this gyral bias. Of course, 0.5 millimeter resolution is potentially going to be very difficult for us to get to in vivo. And so this kind of data can also form as a gold standard for the other approach, which would be to improve the tractography methods themselves. For example, in Oxford, people are interested in trying to develop a model for what a gyrus looks like. And here, if we're going to do this, actually, that makes it even that more valuable if we also have optical imaging that provides a true gold standard. And that leads very nicely to the next topic I want to tell you about, which is how we've been using optical imaging to relate what's happening at the microscopic scale to what we're actually seeing in diffusion. And here, the primary technique that we've been working with is called polarized light imaging. So this is a method that's been pioneered by the Jülich group. I'm sure many of you are used to seeing the absolutely stunning images that they produce. At its most sort of simple form, this is actually a relatively minor modification of a light microscope. You need to add a series of polarizing filters, and one of them needs to be able to be rotated. And what you do is you acquire a series of images at different rotations. And because myelin is birefringent, that means that it interacts with the polarizer in such a way that you get a sinusoidal variation in the transmitted light that tells you the orientation of the fiber. So this is work in my group that Jeroen Molink has been doing. You can produce images like this on the left. So this is a section through the pons of that same brain that I was showing you a moment ago at high resolution. Here, color is coding for direction. So the red to orange part of the color map are fibers that are running superior to inferior. And so here you're seeing the fibers of the corticospinal tract, but you can see interdigitated between them are the pontine fibers, which are crossing between the hemispheres, so they're going in plane. And when we compare this then to the diffusion data on the right, you can see that at this 0.5 millimeter spatial resolution, we're actually starting to capture that interdigitating structure, where we can see the fibers that are going, well, it's essentially crossing over and then going up. Right? So when we first started developing this PLI setup, we thought that we would begin by looking in the corpus callosum because it's a relatively simple, well-known structure in terms of what we would expect the images to look like. And I think this is actually the first polarized light imaging image that we got. And you do see that the fibers are roughly collinear for the lateral aspects of the corpus callosum, but right on the midline, you have this sudden chaotic pattern of the fibers. We thought at first this might be an artifact of cutting or something like this, but we saw it repeatedly on samples in the callosum. And when we then looked in the diffusion imaging data, you actually can see precisely the same thing. And in fact, you can start to see what seems to be happening is that the fibers are actually on the midline, they're sort of braiding relative to each other. So this kind of suggests that the corpus callosum is a little bit more interesting than maybe we thought it was. So we thought we would use this as a test bed for trying to relate microscopy to diffusion signals. And in particular, we wanted to see if we could validate a measure that had been proposed by Stam Soteropoulos for diffusion data, which is trying to estimate fiber dispersion. So this is something that could be a marker in and of itself, but it also is something that could be useful in refining diffusion tractography techniques. So the basic idea is we take the microscopy data, in this case PLI, we create a supervoxel that corresponds to the size of our diffusion data, and then we can use that to create some kind of gold standard fiber orientation distribution. Now, we didn't want to limit ourselves to just PLI because obviously there's a lot of different aspects of microstructure that could be relating to the diffusion signal. So we also acquired myelin stains and glial stains, and we can do the same thing, slightly different processing, but we can extract these sort of distribution functions and from that extract dispersion profiles. So taking this then into one example sample, which is representative of the sort of three that we looked at. First, if we're looking sort of from left to right, you see that in the diffusion MR signal, the dispersion that we estimate is capturing exactly that pattern that we could see visually in the PLI data. High dispersion on the midline, lower dispersion as you move more laterally. Now, if we compare that to the predictions of dispersion based on the myelin stain, we actually see a remarkably good agreement. It's doing quite a good job of predicting the level of dispersion that we have. Now, interestingly, the PLI, which is shown here in blue, underestimates the level of dispersion. And we do have some thoughts on why maybe that would be actually what is happening here. Also, when you see in orange, what we've done is we've added in an additional glial component of astrocytic processes. And here, what's interesting is that that doesn't actually really improve the prediction. If anything, it makes it slightly worse. So this suggests that there is good agreement between the diffusion estimates and the histologically defined estimates. And this isn't just true in the callosum. We can look at other regions, including regions of crossing fibers, the centrum semioval, and also regions where fibers are fanning. And we get a very nice agreement between the diffusion MR data and the histopathology. Now, of course, we started doing this because we wanted to explore these relationships between MR and diffusion, or rather MR and microscopy. But in doing so, we kind of discovered this feature, and as looking around in the literature, we couldn't actually find where this had been described before, this high dispersion on the midline. So we thought we would see whether we could see this in vivo data as well. And so we pulled out our favorite data set, which is the biobank data, and sure enough, you see the same pattern of dispersion in the in vivo data, which was really gratifying. It's kind of using this microscopy data to discover new things that we can then go and look for in our MR data. But Yurin hasn't stopped there. What he's now done is he's asking the question of whether there's any functional consequence of this profile of dispersion. And so he took the biobank resting state data and looked to see if functional connectivity between homotopic regions that are connected via the callosum, whether the degree of functional connectivity relates to the region of the amount of dispersion in the region of callosum that connects those regions. And in fact, he found a number of areas where this seems to be the case. And in particular, he did quite a nice job of doing a split group retest, where a number of these brain regions also survived the group retest, and that's what I'm showing you here on the left. So this is a really nice example of kind of closing the loop between microscopy measures, postmortem imaging, and in vivo imaging. Now, in the long run, with looking at relationships between optical imaging and histology, what we're really aiming to do is to move beyond just a one-to-one comparison between, for example, diffusion imaging and myelin stains. This is kind of the classical way we've tended to go about things in MRI, but it is somewhat limiting because we know that MRI signals, almost every MRI signal, is not sensitive to only one aspect of the microstructure. MRI signals are very sensitive, but they're also very messy. And so what we want to do is actually just say, well, why don't we just embrace this complexity and estimate complex relationships between many MRI signals and many histopathological signals. And we believe that postmortem MRI as an intermediary is going to be absolutely critical to making this happen because we can have not just common tissue state, but common tissue samples for estimating these relationships on the bottom. So we're basically undertaking now a very large data gathering exercise, which will hopefully enable us, for example, to use machine learning techniques to estimate these relationships. And once we've done that, what we have is a tool that can take in vivo MRI data and predict a variety of histopathological stains, what you would see in the brain if you were able to do histopathology in vivo. Okay, so it would give us a very unique view into the brain. So finally, I want to just, in terms of spatial scales, I just want to tell you briefly about what we're doing at even higher resolution, which is electron microscopy. And this work's a little bit newer, but the goal of this work is to really address one of the challenges of biophysical modeling, right? Which is that you always have to make assumptions. And it's hard to be confident that you've made the right assumptions if all you ever do is compare a signal prediction to the signal that you've measured. So what we want to do is to be able to characterize in great detail what the actual underlying microstructure looks like. So if we've measured the underlying microstructure and we can predict the right signal, then that gives us greater confidence than if we had made assumptions about what the microstructure looks like. It can also help us to refine which assumptions we can and can't safely make. So this is work by Michiel Klein-Einhaus. And what he's been doing is using what's called a block face EM scanning technique. So you scan the face of a very tiny block of tissue and then use a diamond knife to just shave off a very thin upper layer. And by doing that, you then build up a 3D EM data set that is intrinsically very well co-registered. This is one of the hardest things to do if you have a series of slices, is getting them to co-register. But because it's a block face technique, they're already well registered to each other. Now, typically with EM data, a lot of the segmentation that one might do to define compartments is still done by hand. And there's just simply no way that you could roll that out in a 3D volume of this. It would just take years and years to segment. So what Michiel's been doing is focusing on coming up with a semi-automated segmentation pipeline, where he can define now with a substantial reduction in sort of burden of manual labor. He can define a number of different compartments. So here I'm showing you the myelinated axons, the interaxonal space, and the myelin sheath, but he's actually got a number of other compartments that he can extract as well, including things like mitochondria or the nodes of Ranvier. So just to kind of demonstrate that this worked, but also because this is a source of information in itself, he can then go in using his segmentations and do things like extract axon diameters from all the myelinated axons he's segmented, or the unmyelinated axons. And you can see here that the axon diameter of the unmyelinated axons is in general smaller, which is exactly what we'd expect. So that gives us some confidence that it's doing something sensible. And on the right, you can see that he's defined a near linear relationship between the myelinated axon diameter and the G ratio, which is the thickness of the myelin sheath. But in the long run, what we really want to do with this is to do highly realistic simulations of signals, in particular diffusion signals, where he, based on his segmentation, can define meshes, and he can then insert diffusing water molecules into these different spaces, each with different properties, and form then a highly realistic simulation of what diffusion looks like. And from there, it's actually a very well-known problem how you can turn that then into a signal prediction of diffusion MRI. And so ultimately where we think we want to use this kind of approach is, having built this 3D model, we would validate it using rodent experiments. So, for example, with Heidi Johansenberg, we're very interested in looking at plasticity, and in particular using transgenics to modulate whether myelin exhibits plasticity or not. Having then validated these models, we have something that we basically have something we can use to do in silico manipulations of these compartments. And that's potentially a really powerful thing to do. It enables us to manipulate compartments in ways that we can ask questions about what aspects of the microstructure are central to the diffusion signal, and what signals could we maybe try to extract that would make us more sensitive to certain changes in the underlying microstructure. And ultimately then that would provide us with novel markers that we could roll out in vivo. So in the last few minutes, I'm just going to talk about population scales. Now, I'd encourage everyone to, if you're interested in this, please check out the symposium this afternoon. You're going to hear about a number of studies along these lines. I'm just going to talk about the study that I'm involved in, which is the UK Biobank. So the Biobank, as many of you know, is an existing epidemiological cohort of 500,000 individuals. They've already gone extensive lifestyle surveys, biological assays, genotyping, and crucially, we have long-term health outcomes that will be tracked by the National Health Service in the UK. And we're now bringing back 100,000 of those subjects for imaging. And it's really kind of hard to conceive of what 100,000 subjects looks like. So I'm going to show you a gratuitous animation. So this is maybe what your typical study looks like. It's no small task to acquire this kind of a data set of this size. People have begun to do more ambitious studies. So we now have a number of studies that are imaging in the order of thousands. But if you compare Biobank to that, it's absolutely enormous. It's stunning, okay? And the reason why we need this data set is because we're scanning individuals before they get sick, because we want to be able to find that needle in a haystack of individuals who go on to develop motor neuron disease, or other individuals who go on to develop Alzheimer's disease, because this gives us a really important window into early markers of disease. So one of the main ways that we can envision this data set being used, although of course it's not the only way, is that we have our imaging data along with a rich variety of other types of data with health outcomes that occurred then later on in life. So we can learn the relationships between these measures and the long-term health outcomes, and then begin to predict in new individuals using the kind of measures that can be easily derived in a clinical setting. We can learn to predict what their long-term health outcomes are likely to be. So we only have now 35 minutes in order to achieve this very high number of subjects. And so it was a real challenge to set up the protocol. This is basically my role in the study. So we have managed to squeeze in quite a few modalities because we don't want to be specific to any particular disease. So in addition to, of course, T1 weighted images, we're getting T2 flare for lesion detection. We have diffusion scans for tractography and microstructural imaging, resting state scans for functional connectivity, task fMRI, and susceptibility-weighted imaging. And I do just want to say how important the simultaneous multi-slice techniques in both diffusion and fMRI have been for this study, because they've really enabled us to get this amount of data without having to accept high levels of compromise for any given modality. So just taking a first look at this data, so this is the first 5,000 subjects. We were able to extract, so Fidel in the FSL group was able to extract 2,500 imaging phenotypes. So these are things like volumes of certain structures or the certain microstructural markers in particular white matter pathways. And we then compared this using simple univariate correlations to 1,100 non-imaging phenotypes. So that, if you multiply those numbers together, that gives you 2.8 million univariate tests that we did, okay? Which is not the sort of thing that we've generally been able to sensibly do in imaging up until now. But even with full multiple comparison correction, for example, with the FDR correction, we're able to get about 30,000 pairwise associations. If you do Bonferroni, very conservative correction, you get 10,000. We've never been in this kind of a domain before where we're able to do this kind of GWAS analysis, and it's purely about the number of subjects. And this is just the first 5% of the ultimate study. Now, of course, it's oftentimes difficult to interpret a single univariate correlation, let alone 30,000 of them. So one of the directions that we actually think is going to be most useful in the biobank data is to do things like identify population modes. So this is an intrinsically multivariate approach. And the potential for this is really nicely illustrated in this paper by Thomas Insel, where he was thinking about psychiatric illness and the fact that currently we typically think of diseases in terms of symptomatic categorization. But we know that that's problematic. We know that sometimes multiple biological sources could lead to the same symptom, and similarly we know that several symptoms could have the same underlying biological source. And so the idea here is that if we have very rich data sets to work with, we can move away from that and let the data tell us what is happening in individuals by simply looking to say, how can we cluster individuals into groups, hopefully identifying more biologically meaningful characterizations than symptoms can provide. So one way we can do that is using what's called canonical correlation analysis. So basically what we're doing is we're looking to identify common patterns between two sets of variables. So here, for example, we might have one data matrix that's subjects by imaging measures and another that's subjects by health outcomes. We want to find common patterns in those data sets. And so what CCA does is very similar to ICA. It finds ways of combining the imaging measures and separately a different way of combining the health outcomes such that you get a common mode of population variation. And it can be, when you look at these data matrices, it can look a little bit magical, but so if I just resort the data according to the strength in the CCA mode, you see that there were patterns there that existed in those matrices. Some of the imaging measures have this pattern across it, some of the health outcomes have these patterns across it. Okay? So this is a way of trying to pull out these patterns. Now, when we did this in the biobank data, we were able to identify about 9 modes of variation that were statistically significant. They're kind of difficult to explain quickly, so instead I'm going to show you where we did the equivalent analysis in the Human Connectome project. So this was data that was obviously a very big group effort as part of the Human Connectome project. But Steve Smith took the functional connectivity data. So that's our sort of subjects by imaging measures on the one hand, and then a whole host of lifestyle factors that were gathered on the other hand. And he did this canonical correlation analysis, and a single mode emerged from that. And it was actually remarkably interpretable. So it's the series of connections shown here on the left, but I'll just draw your attention momentarily to the right. So these are the sets of individual phenotypes or lifestyle factors that came out as having a significant contribution to the CCA mode. And as you look through the list, so they're sorted here by how strongly they contributed and whether they contributed with a positive or negative coefficient. And if you look, what you see is on the upper end, the positive end, you have basically positive lifestyle traits. You have things like level of education, socioeconomic status, fluid intelligence. If you look on the negative end, you see what we commonly think of as negative lifestyle traits, drug taking, risk taking, that sort of thing. So what this mode describes then are a series of brain connections that link to these different life factors. And in particular, if you score highly on this CCA mode, so you have largely good lifestyle factors, then your connections have high functional connectivity, and vice versa. If you score low on this mode, where you have more negative life factors, you have lower levels of connectivity. So it's sort of suggesting that there's an imprint on the brain of these sort of life factors. And of course, we can't infer causality, but nevertheless, we see that there is a very clear, if you will, biological aspect to these life factors. So with that, I would like to just conclude by talking a little bit about some of the themes that have come up in my talk today. The first is that I think we need to take very seriously the goal of bridging scales and bridging technologies. And bridging technologies not only includes doing multimodal imaging, but also doing things like taking information or what we know about data based on image analysis and incorporating it at an earlier stage in order to condition the data to be well appropriate for that analysis. We need to improve our biological specificity. I think it's one of the biggest challenges that we face in MRI is that our signals are sensitive, but they're not specific. And that relates very strongly to this idea of bridging scales and technologies. It's, of course, a catchphrase, but certainly with population imaging as well as various other senses in which we are collecting large data sets, we're moving very much in the direction of big data. It's going to change what we can do with our techniques, but it's also going to have a lot of challenges associated with it. Finally, I think we need to think about a new kind of scientist. All of these goals that I've described require people to move outside of the sort of silos that we have defined in terms of how we train people currently. And in order to leverage the human potential that we have in societies like this, we need to be very thoughtful, I think, about what's the right way to address these challenges in terms of creating people who are able to address these problems. Sometimes that may mean that we need to create fundamentally cross-disciplinary scientists. Sometimes it may mean that we just need to have a culture change in the way we think about collaboration. But I think it's very worthwhile having this conversation, particularly in gatherings like this. So with that, I would like to thank you for your attention, thank my group, in particular these 5 individuals whose data I've been showing you today. Thank you.